This next video is going to be on cell division, cell diversity, and cellular organization, which is basically mitosis and meiosis. So, what is the role of mitosis? To produce genetically identical cells, therefore mitosis does not give rise to genetic variation. The cell cycle. So in mitosis, there are 4 stages: prophase, metaphase, anaphase, and telophase. In cytokinesis, the parent and replicated organelles move to opposite sides of the cell. The cytoplasm divides, thus producing 2 daughter cells. And in interphase, the cell grows and prepares to divide. Chromosomes and some organelles are replicated. Chromosomes also begin to condense. So in the process of prophase, there's 4 points that you need to know. The nuclear envelope breaks down and subsequently disappears. That's the first point. Number 2, chromosomes condense. Number 3, centrioles move to opposite poles of the cell. And part 4, for the purpose of spindle formation. Then in metaphase, the chromosomes move to the equator and attach to spindle fibers via centromeres. And in anaphase, the sister chromatids separate. And in telophase, the nuclear envelope reforms, creating 2 daughter cells. The spindle is broken down and disappears. The chromosomes uncoil. Then in meiosis, this process of meiosis, the interphase part. So during interphase, the cell's DNA unravels and replicates to produce double-armed chromosomes called sister chromatids. So meiosis includes 2 divisions, meiosis 1 and meiosis 2. Meiosis 1 is the reduction division. It halves the numbers of chromosomes. So what happens in prophase 1, the chromosomes condense, getting shorter and fatter. Homologous chromosomes pair up, so number 1 with number 1, 2 with 2, blah, blah, blah. Crossing over occurs. Centrioles start moving to opposite ends of the cell, forming spindle fibers. The nuclear envelope breaks down. In metaphase 1, the homologous pairs line up across the centre of the cell and attach to the spindle fibers by their centromeres. Homologous pairs line up across the centre of the cell and attach to spindle fibers by their centromeres. In anaphase 1, the spindles contract, pulling the pairs apart. 1 chromosome goes to each end of the cell. In telophase 1, a nuclear envelope forms around each group of chromosomes. Cytokinesis, which is the division of the cytoplasm, occurs and 2 haploid daughter cells are produced. So then meiosis 2 occurs. So the 2 daughter cells, undergo prophase 2, metaphase 2, anaphase 2, and telophase 2, and cytokinesis. These are pretty much the same as the stages in meiosis 1. except with half the number of chromosomes. In anaphase 2, the sister chromatids are separated. Each new daughter cell inherits one chromatid from each chromosome. 4 haploid daughter cells are produced. So then genetic variation is the differences that exist between individuals' genetic material. So the reason meiosis is important is because it creates genetic variation. It makes gametes that are all genetically different. So basically mitosis produces genetically identical cells and this produces genetically different cells. So creating genetic variation in gametes occurs in 2 different events. The first is crossing over of chromatids. So during prophase 1 of meiosis 1, homologous pairs of chromosomes come together and pair up. The chromatids twist around each other and bits of chromatids swap over. The chromatids still contain the same genes but now have a different combination of alleles. The crossing over of chromatids in meiosis 1 means that each of the 4 daughter cells formed from meiosis 2 contains chromatids with different alleles. And then number 2 is independent assortment of chromosomes. So each homologous pair of chromosomes is 1 chromosome from your mum and 1 chromosome from your dad. When the homologous pairs line up in metaphase 1 and are separated in anaphase 1, it's completely random which chromosome forms each pairs and which ends up in the daughter cells. So the 4 daughter cells produced by meiosis have completely different combinations. This is called independent assortment